So this slide is going to talk to us about the overall structure of a piece of DNA. So first of all, we have our individual nucleotide that we've already met. It's a nucleotide. It has a phosphate, a 5-carbon sugar, and a base. So it's following all the rules that we've already learned. That sugar can be either ribose or deoxyribose, depending on what type of molecule we're building. Are we going to build DNA? Are we going to build RNA? And then the base, that can be any one of these things, a C, a T, an A, or a G. Or if you're building RNA, you use U instead of T. So this is our basic nucleotide building block. And then we take these building blocks and we hook them all together in a long line. And how they're hooked together is kind of interesting. So here are 1, 2, 3, 4, 5, 6 nucleotides all hooked together in a long chain. And when that happens, you can see here that this is a phosphate, the sugar, and a base. This is the first nucleotide in the chain. Here's the next one: phosphate, sugar, base. Here's the next one: phosphate, sugar, base. So what we've created here by hooking these together is a length of nucleic acid built of 6 nucleotides. And you can see there's what we call the phosphate sugar backbone. You see how it's basically phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. Those are all making the long chain down here. And we call that the phosphate sugar backbone. And then the bases are kind of sprouting off from the backbone, kind of like the teeth of a comb. So this is like a comb. This is the rigid part, the spine of the comb. And here are all the bits, the teeth that are sticking off of it. And on this diagram, you can see this is a purine. This would be a pyrimidine. We don't know which one, but we know it's definitely a pyrimidine because it's a single hexagon. Here's another pyrimidine. Here's a purine. Here's a pyrimidine. Here's a pyrimidine. So what we can see has happened here is that we attach the sugar of one nucleotide to the phosphate of the next. And then this sugar is attached to the phosphate of the next, the phosphate of the next. So that's what we're attaching, is the phosphates to the sugars all the way down this chain. So that's a single strand. If we go to this slide here, we can see, I move myself over. All right, so here once again is our phosphate sugar backbone. Phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar coming down here. And here are the bases sprouting off from the first carbon one of each sugar down here. Phosphate sugar backbone, bases sprouting off like the teeth of a comb. And then if we look on the other side, there's another chain on this side, phosphate sugar, phosphate sugar, phosphate sugar, going up and down here with these bases sprouting off from carbon number 1. Here's a purine, a guanine. Here's a cytosine. Here's an adenine. So this is what we would call a DNA molecule. Many textbooks would call this a DNA molecule. It's not strictly, though, a single DNA molecule. Most textbooks kind of say that wrong. Remember, for something to be considered a molecule, it has to be connected together with covalent bonds. So if we look down here, yes, this is all connected with tons of covalent bonds, lots of solid lines hooking this together. And if we look on the other side, there's covalent bonds hooking this all together. But if we look in between the two sides down here, hooking between the left side and the right side, there are no covalent bonds. These dotted lines represent hydrogen bonds holding the left side to the right side. So we can't call this a single DNA molecule. This is actually 2 DNA molecules that are lined up facing each other. And it's more accurate to say that DNA is a double-stranded molecule. It's made of 2 strands or 2 sides. And what you can see happens here is this one side and the other side are paired up according to rules, where if you have a T on this side, on one side, you have to have an A on the other side. A always pairs with T, and C always pairs with G. And that is a absolute hard and fast rule. You can't break it. If there's A on one side, you've got to have T on the other. If you've got C on one side, you've got to have G on the other. A purine pairs with a pyrimidine each time. All right. Now, the other thing to know about this diagram is the ends of the chains. So if we look at this side here, this strand on the left side of this piece of DNA, flapping in the breeze up here is a phosphate group. All right. And this phosphate group is attached to carbon number 5 because phosphate groups are always attached to carbon number 5. And so we call this the 5' end of the DNA. This is somewhat similar to, if you were to read in English, you kind of learn the rules of the language where you start something with a capital letter and you write the sentence, then you end the sentence with a period. And that way and we always read from left to right. And that's kind of like the rules of how we know how to decode the English language. Start with a capital letter, read from left to right, end with a period. So we have a directionality to our language when you see it written down. And that's the same with DNA. Because this DNA is actually a language, it's got a code hidden in it. You have to know the direction to read it. And so to make one end different from the other, like a capital letter and a period, we have the 5' end, which is where the phosphate group is flapping in the breeze. And if we follow all the way down to the other end, down here flapping in the breeze, we have the hydroxyl group that's attached to carbon number 1, 2, 3. If you remember on one of the other videos, we said that carbon number 3 always has a hydroxyl group. All right, and so we call this the 3' end because this thing that's flapping in the breeze is attached to carbon number 3, and it's always going to be an OH or a hydroxyl group. So looking at this side of the molecule, you've got a 5' end to 3' end. We go 5 to 3 going down the slide. This we would label as a 3' end. Looking on the right side here, what kind of end do we have here? Well, flapping in the breeze, there's a phosphate group. Phosphate is attached to carbon number 5, so this is a 5' end right here. And if we follow all the way up and find the other end, what's up here is an OH, and that's attached to carbon number 3. So this is a number 3 or a 3' end. So this is another hard and fast rule for DNA. That you'll have a 5' end and a 3' end for each strand, and they always will be opposite to each other. So if you go 5 to 3 on this 5 to 3, you're going to go 5 to 3 the other way on the other side. They run opposite each other, and this is called being anti-parallel. So you may have heard a statement that DNA is anti-parallel. All that's saying is that one side goes down from 5 to 3 and the other side goes up from 5 to 3. And being able to identify those 5' and 3' ends is really important. So remember, you're just looking for what's flapping in the breeze at the end. Is it an OH? In which case it's a 3' end. Is it a phosphate? Oh, in which case it's a 5' end.